Welcome my colleagues and my students, and today we are completing the topics of autonomic nervous system, and we are speaking about the chemical transmission in the autonomic nervous system. As a general rule, we have two types of transmission inside the nervous system. The first, what is called the chemical transmission. The second is what is called electrical transmission. These are the two methods of transmission inside our nervous system. Today, we are speaking about the chemical transmission inside autonomic nervous system. However, in the coming lecture, we will discuss the process in detail and we will speak about all types of transmission, which includes also the electrical transmission. What is the synapse? Synapse is the site of contact between a neuron and another cell. It is a site where impulse travels from a neuron to another cell. The other cell is classically another neuron, but it may be a muscle cell or a gland cell. So the fate of the neuron is to terminate either on a muscle cell forming neuromuscular junction, or on a gland forming neuroepithelial junction, or on another neuron forming a neuroneural junction, which is the classic form of synapse. This is the first neuron, this is the second neuron. The impulse is transmitted here in this area. This point is called synapse. Any site in which impulse is transmitted, it is called synapse, whether the second cell is a neuron or a muscle cell or even a gland cell. Classically, the chemical synapse consists of three portions. The first is the last part of the first neuron. The second is the space between the two structures. The third is the first part of the second cell, whether it is a nerve cell or a muscle cell. So we have pre-synaptic membrane, then the synaptic cleft, which is the space between both, and the third is the post-synaptic membrane. This is the classic structure: the presynaptic membrane, the synaptic cleft, and the post-synaptic membrane. For more details, this is the presynaptic membrane as we see. This is the cleft. This is the post-synaptic membrane. The details of the process of transmission will be detailed later on in the CNS. However, here we will take a simple idea about the process of transmission. The main components of the presynaptic membrane are the vesicles which contain the chemical transmitter. Classically in the autonomic, we have acetylcholine and noradrenaline, and sometimes we have dopamine and some rare transmitters. Also in the presynaptic membrane, we have the voltage-gated calcium channels, and these channels are important because these channels are the sites where calcium enters the cell on arrival of the nerve impulse. On the postsynaptic membrane, we have important structures which are the sodium channels, which open by binding to the chemical transmitter during the process of transmission. To simplify the process of transmission, when the impulse arrives to the nerve terminal, this causes opening of the voltage-gated calcium channel. So calcium enters the presynaptic membrane and binds to vesicles. Then it moves the vesicles towards the membrane where they rupture and release the chemical transmitter inside the synaptic cleft. Later, the chemical transmitter crosses the synaptic cleft and binds to the postsynaptic receptors and opens them, causing sodium influx. The influxed sodium causes depolarization, which causes initiation of impulse in the postsynaptic membrane. This is a simple description for the process of transmission, and the details will come later. We have two types of transmitters: acetylcholine and noradrenaline. Transmission that is dependent on acetylcholine is called cholinergic transmission. That is dependent on noradrenaline is called adrenergic transmission. So we have also two types of fiber. Fibers releasing acetylcholine are called cholinergic. Those releasing noradrenaline are called adrenergic. Also, we have two types of receptors: cholinergic receptors for acetylcholine, adrenergic receptors for noradrenaline. Later, we have important topics regarding the chemical cholinergic transmission, which is one of the main topics in the transmission inside the autonomic nervous system. We have two topics, cholinergic transmission and adrenergic transmission. Cholinergic transmission is a transmission that is dependent on acetylcholine as a chemical transmitter. It is widely distributed inside our body. Understanding the principle of cholinergic transmission is so essential in all medical specialties. It is life-saving subject. It is not simple. It enters in all specialties. You must understand well what are the cholinergic receptors, how these receptors work, what are the activators, and what are the inhibitors. This is so vital for your clinical life later on. The first important topic in cholinergic transmission is to know the sites where acetylcholine work as a chemical transmitter. First is all parasympathetic fiber. As we know, the course of autonomic, whether in the sympathetic or parasympathetic, consists of preganglionic, then the ganglion, then the postganglionic. The chemical transmitter inside the parasympathetic, whether in the ganglion or the postganglionic, is acetylcholine. So that acetylcholine is the official transmitter of the parasympathetic nervous system and no exception. The second side is the sympathetic fiber to sweat glands and blood vessels of skeletal muscle. The official transmitter of the sympathetic is noradrenaline in most sites except two sites, sweat glands and blood vessels of skeletal muscle, where in these areas sympathetic transmits by acetylcholine. So the nerves supplying these structures are called cholinergic sympathetic fibers, not adrenergic as the general rule in the sympathetic. The third is the autonomic ganglia. The autonomic ganglia, whether sympathetic or parasympathetic, transmit via acetylcholine. So all preganglionic fibers, sympathetic and parasympathetic, release acetylcholine. The fourth site is so essential, so important, neuromuscular junction for skeletal muscle. This is so essential and it has many important clinical applications. The fifth is some synapses in the CNS. All transmitters are present inside the CNS. Please remember these 5 areas and in sequence. You must remember them in sequence. You know the first, the second, the third, and the fourth, and the fifth in this sequence. Later, what about the synthesis of acetylcholine? Acetylcholine is synthesized in the presynaptic nerve terminals, just beside the vesicles, by a simple reaction, simple one-step reaction in which acetyl coenzyme A binds to choline in the presence of choline acetylase, which transmits acetyl group from acetyl CoA to bind it to choline, forming acetylcholine and coenzyme. This is a simple, fast reaction that occurs inside the presynaptic membrane. After doing its action, as any transmitter inside our body, it has to be removed. The main methods of removal are diffusion and hydrolysis. Diffusion means that the molecules are distributed everywhere, so their concentration decreases. However, hydrolysis is the main process of removal. It means that the molecule is broken down by an enzyme that is called cholinesterase, and this enzyme is so important in clinical life, where there are many drugs, there are many toxic conditions depend on this enzyme. And we have many applications that will be discussed later on over the cholinesterase enzyme. When it works on acetylcholine, it breaks it down into choline plus acetate or acetic acid. An important topic in the cholinergic transmission is the actions of acetylcholine. We have two patterns of actions. Scientists noticed that in some sites acetylcholine work like a slow transmitter that has a slow onset and long duration. However, this same transmitter in other areas, it works as a very fast transmitter that works within short time. They concluded that acetylcholine has two main actions that are different from each other, but every one of them has its own sites, its characters, and its receptor. The first one is the slow one. It is called muscarinic action because it is first discovered using a substance called muscarine, which is derived from mushroom. The second is called the nicotinic, which is the fast action, and it is called nicotinic because of the use of nicotine in its description in the first times. The muscarinic action is slow in onset, but it has a long duration. The nicotinic action is rapid in action and it has a short duration. The second is so important where these actions are present. Muscarinic actions are present in place 1 and place 2. Try to remember, place 1 was all parasympathetic fiber. Place 2 was sympathetic to sweat glands and the blood vessels of skeletal muscle. Nicotinic action is present in place 3 and place 4. Place 3 is the nicotinic. Place 3 is the autonomic ganglia and place 4 is the motor in the blade. We have two actions, so we have to compare the receptors of both the muscarinic receptor and the nicotinic receptor. And the first aspect of comparison is the site. The muscarinic receptor present in all parasympathetic, place 1 and place 2 as we said. The second site is the sympathetic to sweat gland and the blood vessel skeletal muscle. The nicotinic receptors are present where nicotinic action is present. In place 3 and place 4 is the nicotinic ganglia and in the neuromuscular junction. The stimulus for muscarinic receptor is the muscarine, which is derived from mushroom and which was first used for discovering these receptors. However, the stimulus for nicotinic receptor is a small dose of nicotine. The blocker for the muscarinic receptor is famous, which is the substance atropine. The blocker for the nicotinic action is large dose of nicotine. For more details, you can read about the types of muscarinic receptors, which are M1, M2, M3, and M4 and M5 receptors, and you can know well the site of each receptor and you can know well about the function of each one. We reached the end of cholinergic transmission. Later on, we will speak about the pharmacology of the cholinergic transmission. See you again and goodbye.